The primary visual cortex, V1, part 1. From retina to V1, retinotopic mapping. In this figure, we see that signals from the two retinas are sent to the central brain, first to the primary visual cortex, also called V1. The lateral geniculate nucleus is the brain region en route to V1. Here is a schematic for the visual pathway from the retinas to V1 via LGN. And this LGN has been seen as a relay station because the resistive fields of the LGN cells resemble those of the retinal ganglion cells in anesthetized animals. However, since LGN receives massive feedback from V1, it is expected to function much more than just a relay station. But not enough is known about LGN to have a consensus about its function. If we divide the brain into its left and right half, then we see that signals from both eyes project to each half of V1, and objects in one half of the visual field project to the other half of V1. So each half of V1 receives input from just half of the visual field. Here is half of a visual field, and here is the V1 surface in a hemisphere of monkey's brain. This is the center of the visual field projecting to fovea. Eccentricity increases to the peripheral parts of the visual field, measured by visual degrees of visual angle. And so this half ring is at 20 degrees of visual angle eccentricity, and this is at 40 degrees, and this is roughly 80 degrees in a very peripheral part of the visual field. This shaded region can only be seen in one eye, so this is a monocular visual field region. And this is the azimuth of the visual field going from 0 towards 180 degrees. V1 neurons have small resistive fields, and neighboring V1 neurons have their resistive fields at or near the same location in visual space. And this transformation from the coordinates in the input image to those on the V1 cortical surface is called the retinotopic map. This line going from fovea to this peripheral location is called the horizontal meridian, and it's mapped to this curve on V1 cortex, and this line to this curve. So therefore, eccentricity maps along this direction, x on the cortex. This azimuth curve at 40 degrees eccentricity is mapped onto this curve on the cortical surface, but this azimuth curve at 10 degrees eccentricity is mapped onto this curve on the cortical surface. So azimuth is mapped along this direction, y, on the cortical surface. Therefore, sometimes the retinotopic mapping is called complex logarithmic mapping. This retinotopic mapping expands this tiny fovea to a huge expanded cortical surface area, but shrinks down a large peripheral visual field region to a small cortical surface area. And the amount of cortical surface area devoted to each unit visual field area is called the cortical magnification factor. And this factor decreases quickly with eccentricity E. So therefore, V1 devotes much more resource to the central visual field, very much like the retina devoting more cones to the central visual field.